My name is Mike Pluth, and I'm an associate professor of chemistry at the University of Oregon. My research group focuses on building small molecules, primarily with a purpose toward applications. We like to build small molecules that can be used to look at different biological processes. I've become interested in hydrogen sulfide because I have a long-term interest in understanding how small molecules react with different important targets. And one emerging area where this is quite important is in biology, where there are a lot of very small signaling molecules that are important. And H2S is one of the more recent of these, and it's recently attracted a lot of attention, and I think is a very nice system where chemists, biologists, medical professionals, and physiologists can all work on similar problems from different perspectives. Much of the work on H2S in biology is very interdisciplinary, so it requires chemists, biologists coming together with different backgrounds and different areas of expertise. And so certainly as a chemist, we don't do a lot of biological work, and as biologists, they aren't going to do a lot of chemical synthesis. So this is one place where we can work together and work on big problems where each of us brings our own expertises. I think that one of the long-term potential applications of our research in a broader context is toward human health. And because H2S is very important in biology, if we can understand the exact mechanisms by how it works, we can really engineer new technologies or medicines or therapeutics that might have a large impact in a lot of people's lives. One thing that excites me about this line of research is that often the results are fairly unpredictable, and what worked on paper may not work in practice. And so trying to unravel what's actually going on and the chemistry that's occurring is quite exciting. One of the important aspects of our research is being able to visualize where hydrogen sulfide is being produced. And to do that, we often use a fluorescence microscope, which allows us to see the fluorescence emitted by these small molecule probes that we create in real time in live cells. One big challenge of working with hydrogen sulfide is that in many contexts, it can be very sensitive to both oxygen and water. And so with model systems, we work in glove boxes that allow us to work with this reactive molecule in a very isolated environment. Every student is an individual, and that what works to connect with one student may be different than another. And that's true in the classroom and in the research lab. And also, you learn a lot from the students as a teacher. And so it's really a two-way street where a question that a student might ask really makes me think about things in a new way.